The citric acid cycle, also known as the Krebs cycle, is the second phase of cellular respiration. It happens after glycolysis but before the electron transport chain. The Krebs cycle takes place in the matrix of the mitochondria, the cell's powerhouse, and its purpose is to break down absorbed pyruvate in order to create ATP. Pyruvate supplies the body with pyruvic acid, which plays an important role for manufacturing and using energy. When those two pyruvate enter the matrix of the mitochondria, the Krebs cycle begins. The cycle is a series of 8 reactions. The first reaction starts with pyruvate and is converted into acetyl-CoA. Acetyl coenzyme A is used to begin the cycle and is used to deliver the acetyl group to oxaloacetate, which with the 2 carbon acetyl groups combines to form citric acid. This conversion happens by the action of 3 enzymes. Such enzymes are the enzymes that produce molecules such as NADH or FADH2. Accordingly, out of this reaction, an NADH molecule is produced. Notice that the acetyl-CoA is only a 2-carbon byproduct, yet pyruvate contains 3 carbon molecules. This is because one of the carbon molecules is used up in order to convert the pyruvate into acetyl-CoA. The next cycle, when a 4-carbon molecule known as oxaloacetate, which is a crystalline organic compound that is formed by the oxidation of malic acid, an acid formed out of the hydration reaction of an alkaline to form an alcohol, and is acylated from citric acid in the citric acid cycle. In this cycle, oxaloacetate combines with acetyl-CoA to produce a 6-carbon molecule known as citrate. Citrate is a derivative of the citric acid, which is primary to the citric acid cycle. Next, citrate becomes a 6-carbon molecule and is called cis-acetonate, which then becomes a 6-carbon molecule isocitrate, which is commonly used to test the authenticity of fruit products. In this next step, we lose 1 carbon molecule as carbon dioxide, and we also produce another NADH. NADH is the reduced form of NAD+, and it can be used to donate electrons. These electron transfers are the main function of NAD and NADH, although they are used to add or remove chemical groups from protein. After we lose 1 carbon and produce another NADH, we are left with a 5-carbon molecule named alpha-ketoglutarate. In the next step, we once again lose a carbon molecule to carbon dioxide and gain NADH. Carbon dioxide is composed of 2 oxygen atoms bonded to 1 carbon atom. In the Krebs cycle, carbon dioxide is released, which we breathe out. Back to the cycle. Therefore, after losing a carbon and gaining an NADH, we are left with a 4-carbon in our main molecule, which is now called succinyl-CoA, a combination of acetyl-CoA and succinic acid, an acid that serves as an energy-yielding process in the TCA. Succinyl-CoA is then converted to succinate, and a GTP, guanosine triphosphate, is produced in the process. GTP is a source of energy and an activator of substrates in the metabolic reactions. It is used as a source of energy for protein synthesis and is formed by the GDP-forming succinyl-CoA synthetase. It may be utilized to form ATP. Succinyl coenzyme A synthetase is the enzyme that catalyzes the reaction between succinyl-CoA and succinate. The enzyme facilitates the coupling of this reaction to the formation of a nucleoside triphosphate, either ATP or GTP, from the formation of an inorganic phosphate molecule in either GDP or ADP. It plays a key role in one of the major catalysts involved in the citric acid cycle. The next step begins when succinate is then conformed to fumaric acid and an FADH2 is produced in the process. Fumaric acid is one of the 2 isometric unsaturated dicarboxylic acid, the other being malic acid. Fumaric acid is the result of the oxidation reaction of removing hydrogen from saturated carbons to form an alkaline fumaric acid. The FADH2 molecule that is produced is a form of flavin adenine dinucleotide. It is involved by several important reactions in metabolism and can convert from FAD to FADH2, its 2 forms, by accepting or donating electrons. FADH2 is significantly higher in energy than FAD. Fumarate becomes malate, and in the process, H2O is produced. Fun fact, the Krebs cycle uses up 3 water molecules for every pyruvate that enters the Krebs cycle. And malate completes the cycle and becomes acetoacetate, producing the final NADH in the process. The acetoacetate is then readily available to combine with any available acetyl-CoA and begin the process again.